Good morning. We are excited for another day of, for me, great learning and a lot of presentation of exciting research that's going on. So we're starting off with two more fellow presentations. And I'd like to invite Dr. Michael Lidsky to the stage to give our first one today. Dr. Lidsky is an assistant professor of surgery within the Division of Surgical Oncology at Duke University. And his practice focuses on the surgical management of cancerous conditions of the liver, biliary tree, and pancreas. Dr. Lidsky also leads the hepatic artery infusion team at Duke, a rapidly growing program to deliver high-dose chemotherapy directly to the liver for patients with advanced primary and secondary liver cancers, including metastases from colorectal cancer and unresectable intrahepatic cholangiocarcinoma. And he's going to speak to us on improving therapeutic efficacy and durability in FGFR2 fusion cholangiocarcinoma.
Thank you so much, Laura, for the kind introduction. While they're loading my slides, I just wanted to express my sincere gratitude for being included in the conference this year. This is a truly incredible event. I've been really enjoying the time, of course, getting to see old friends and colleagues, but most importantly, making new friends and new colleagues and collaborators. And I think the most amazing part of this few days has been interacting and interfacing with patients and caregivers and advocates. So Stacy, I know you just mentioned you're looking for feedback to make this conference better. It's hard to imagine how you will do that. So thank you. I don't have any disclosures. I do want to begin with a brief mention of gratitude for receiving the Margaret M. Brown Memorial Research Fellowship Award. Margaret immigrated from Scotland to Canada in the 1950s with her husband, Jim. And she had many hobbies, including gardening, baking, golfing, and traveling. But she was also a firm believer in volunteering and instilled in her family how important it was to give back and to work hard. Margaret was stolen by cholangiocarcinoma in 2011 in her 80th year after a strong, dignified, and courageous battle. And this award is in her honor. It's sponsored by Jan, her daughter, and Jan's husband, Dean Meyer, who I had the pleasure of meeting and getting to know last night. As you know, Dean is on the board of the Cholangiocarcinoma Foundation and is here, I think, somewhere in the room today. As you may know, Jan, too, was diagnosed with intrahepatic cholangiocarcinoma in 2018, but is disease-free after surgery and chemotherapy. And Jan and Dean raise money for cholangiocarcinoma research through the annual Quack Out Cholangio 5K, which takes place next month, May 20th. If you scan the QR code on the right, it doesn't get any easier. You can learn more about how to participate, how to donate, and how to get involved, maybe even run. I'd like to thank the Cholangiocarcinoma Foundation and, of course, Jan and Dean for the honor of receiving the Margaret M. Brown Memorial Research Fellowship Award. So over the next 7 to 8 minutes, I'll briefly review the background data that drove the direction of our research, and I will then show you a novel patient-derived model of FGFR2 fusion-positive intrahepatic cholangiocarcinoma that we developed to investigate FGFR signaling, and then potentially identified a new treatment strategy. So as you know, 70 to 80% of patients with advanced biliary tract cancers are not surgical candidates and are left with systemic therapy as their primary and often only modality of treatment. And even with the most recent standard of care regimen of gemcitabine, cisplatin, and durvalumab, survival remains somewhat limited for the majority of patients. Specific to intrahepatic cholangiocarcinoma, this is a single-arm study with pemigatinib, which, as you know, is the first inhibitor to receive FDA approval for intrahepatic cholangiocarcinoma. And in this study, they showed a progression-free survival of 7 months and a median survival that's under 2 years. So given the limitations of the available therapies in this space, we leveraged my unique access to patient tissue as a surgeon and derived a novel model system of FGFR fusion-positive intrahepatic cholangiocarcinoma, and I'll now share with you some of our data. So a patient with unresectable disease who was in the operating room for a pump had given us permission to use his tumor, and that tumor was used to create a xenograft, which is then used to make cell lines and organoids, as you can see in the top of this panel. And then both of those models were reimplanted into mice, which reestablished tumors consistent with that of the patient. To ensure fidelity of our models, we first did immunohistochemistry for both positive and negative markers of intrahepatic cholangiocarcinoma. And you can see on the left side, the xenograft, the organoid, and the cell line were all consistent with that of the patient's tumor. We also performed next-generation sequencing of these models and confirmed retention of an FGFR2 G3BP2 fusion. Now, we first investigated the biological relevance of these models, first looking at FGFR signaling. And in the top Western blots in the cell line and the organoid, when you deprive them of the growth factors and remove serum from their culture conditions, you can see that in the second column of each of those blots, where it says starve, the FGFR fusion is constitutively active, which is indicated by persistence of a band there. And on the bottom blots, when you use pemigatinib as an inhibitor, you can see that FGFR signaling is blocked. So we went on to look at the clinical relevance of these models and first treated the cell line and the organoid with pemigatinib on the left in A, which is the cell line, and D, which is the organoid. You can see that the IC50 for each of these models is in the low nanomolar range. And further, this response is dose-dependent. In panel B, you can see that with increasing dose, you get an increasing loss of viability. And we confirm this with the clonogenic assays shown in C. In the first column, which is the Duke cholangio model, DUC18828, you can see with increasing dose, you get impairment in viability that's dose-dependent. But in two wild-type lines, SSP25 and RBE, there's no loss of viability despite the highest doses. Importantly, we also showed efficacy of the inhibitor in vivo, and that's shown in panel E. Mice that were given pemigatinib, represented by the red line, have an impairment in their tumor growth, which translated into a survival advantage and improvement of 70%, shown in the curve to the right. So additional work looked at the effect of combining gemcitabine and pemigatinib. We first tested gemcitabine alone, and on the left in the cell line and organoid, you can see gemcitabine alone has an IC50 in the low nanomolar range. And on the right, those are synergy array plots, and these can be interpreted as data rises above the zero plane. That's indicative of synergy. Below the plane is antagonistic, and within the plane is considered additive. And I think these plots pretty clearly show that the addition of gemcitabine to pemigatinib is just additive. So in search for a combination that had the potential to augment response to FGFR inhibition, we performed a high-throughput small molecule screen with over 2,100 compounds in the presence and absence of pemigatinib. And on the left in the rank order plot, the further down the y-axis corresponds to increasing synergy. And then we went on to prioritize the drugs on the table on the right side for further validation. So specifically, we found that quisinistat, which is a pan-HDAC inhibitor, in combination with pemigatinib is synergistic in the cell line and the organoid. You can see these plots look quite a bit different than the plots I showed you for gemcitabine, and here the data rise well above that zero plane. We went on to confirm these findings and perhaps the biology behind HDAC and FGFR inhibition here with a different pan-HDAC inhibitor, panobinostat, and when you combine that drug with pemigatinib, again, you see significant synergy here in our patient-derived cholangiocarcinoma model. But most importantly, we confirmed synergy in vivo. So tumor-bearing mice were randomized to control versus monotherapy versus combination therapy. And in mice who were given pemigatinib and quisinistat, which is represented by the green line at the bottom of that tumor growth curve, you can see that there's a statistically significant difference between either of the monotherapies and, of course, the control in a way that is more than additive of either monotherapy. You can see the relative reduction in tumor volume on the right is far greater than pemigatinib plus quisinistat. So we made an attempt to dig into the biology a little bit here and perform flow cytometry under the same treatment conditions. On the left, you can see that in the cell line on top and the organoid at the bottom, there's synergistic impairment and proliferation with the combination therapy. And then on the right, with annexin staining, the combination strategy also augments apoptosis with increased early and late apoptotic fractions. Mechanistically, I think the interplay between HDAC and FGFR signaling is not quite well understood. And in these Western blots on the right, these are some of the compensatory pathways that may change when FGFR signaling is altered. And what you see is that with monotherapy or combination therapy, these aren't altered at all. On the left, if you look at the second column, which is pemigatinib alone, you can see that pemigatinib impairs FGFR signaling, which is consistent with what we had previously shown. Interestingly, though, quisinistat increases FGFR signaling, which was an interesting finding, and that increase is then blocked or impaired with combination therapy shown in the fourth column. And these data led us to hypothesize that HDAC inhibition shifts these models toward an FGFR-dependent state, which can then be more effectively blocked with FGFR inhibition. So in summary, we leveraged patient-derived models to generate novel FGFR fusion-positive intrahepatic cholangiocarcinoma cell line, organoid, and xenografts that are both biologically and clinically relevant. We use these models to identify HDAC and FGFR co-inhibition to be synergistic, and I do think these data justify an early phase trial to investigate not just the clinical relevance, but also the tolerability of this combination therapy in patients. So we published these data late last year in MPJ Precision Oncology, and I'd like to conclude just by thanking once again the Cholangiocarcinoma Foundation and Jan and Dean Meyer for the honor of this award. Thank you.
Thank you, Michael. And we will just now move to the second fellow, Evan O'Loughlin, who is currently a postdoctoral fellow at the Massachusetts General Hospital after he achieved his PhD in Harvard Medical School. And he will give a talk about improving FGFR-driven biliary tumorigenesis and therapeutic response in 3D models.
All right. Thank you for that. Thanks for the introduction. I'm Evan O'Loughlin, and I'm a postdoctoral fellow in Andy McClatchy's lab at Harvard and Mass General. And I'm really grateful for the opportunity to talk to you all today about my research on using on using 3D models to study FGFR-driven biliary tumorigenesis. I have no disclosures. I just wanted to start off by reminding everyone that our body is made up of numerous epithelial tubes, and these tubes can give rise to a number of different forms of cancer. And importantly, the breakdown of epithelial architecture is one of the first signs of tumor onset. And a tube that we're all interested in, of course, are the bile ducts. Here's just a diagram of the liver, and as we all know, the liver is comprised of two epithelial cell types, the bile ducts as well as the hepatocytes, and these derive from a common progenitor called the hepatoblast. And there's two major forms of liver cancer, hepatocellular carcinoma and cholangiocarcinoma, and traditionally it was thought that hepatocellular carcinoma arises from the hepatocytes, whereas cholangiocarcinoma arises from the bile ducts. But recently it's become appreciated that there's actually substantial plasticity within the liver under situations of liver injury, so including liver regeneration, disease, as well as cancer. Again, I'm interested in intrahepatic cholangiocarcinoma, like all of you. And I think one thing that's really interesting about it is that when you look at these tumors histopathologically, you can see that they have this sort of, many of them have this sort of tubular structure to them, which suggests that there's some selective advantage that's being conferred by adopting this sort of organization. And I'm particularly interested in FGFR-driven cholangiocarcinoma. And just to remind everyone, fusions are the most common form of FGFR mutation, and the number of fusion partners have been identified. But there are also other activating mutations, such as mutations in the extracellular domain of FGFR2. In my PhD work, I was particularly interested in the development of the biliary network, in particular how you generate a tube on one side of the portal vein. And to, I guess, summarize a lot of work, I described de novo lumen formation as a central design principle that controls the development of these tubes. Just to summarize with this animation, you can see the formation and expansion of de novo lumen, and this self-organizes a structure until you get a system of ramified ducts. Moving forward, I wanted to move into a more cancer-related space. So I had this kind of question of like whether FGFR mutations that are found in cholangiocarcinoma can aberrantly hijack this process of de novo lumen formation. And to do this, I needed a specific model that enabled me to study lumen formation in 3D. So for this, I exploited a panel of cell lines that were derived by a previous postdoctoral fellow in the lab. So these are embryonic liver cells from a mouse. And these cell lines displayed a whole array of behaviors in 3D. So some cell lines like ELCA made no lumens, and some made like larger lumens, and some made these tubes in 3D. And so I like the system for a number of reasons. I think it has a number of great advantages. One is that it's manipulable with genetic and pharmacological tools. We can culture these cells mosaically to study what factors are autonomously or non-autonomously required for lumen formation. And I adopted the system for quantitative imaging, which I'll get to in a moment. In collaboration with a postdoctoral fellow in the lab, UN, we profiled these cell lines in terms of their expression of well-known biliary fate markers. And through this, we were able to define that the ELCA lines that don't make lumens are more representative of an immature hepatoblast state, whereas the ELCC lines, for example, are more representative of mature biliary cells. And for the remainder of my talk, I'll just focus on this ELCB line. And in collaboration with Shannon Stott's group, we established a quantitative pipeline for studying these. As opposed to a confocal image where you can only quantify a subset of lumens, we did whole slide scans followed by an image analysis program. And this enabled us to quantify many, many lumens, many-fold more than would be otherwise available. I'm going to skip through these next few slides just in the interest of time, but they're in the program, and if anyone is interested in talking to me about it, I'd be happy to do that. So our question was what different mutated forms of FGFR do to impact lumen formation in this assay that I've developed. So here we exploited some previously published mutations in FGFR2 that are associated with cholangiocarcinoma, so an FGFR2 PHGDH fusion, and two mutations in the extracellular domain. And when we look at these on a blot, we can see that the wild-type FGFR2 is present as both a glycosylated species, which I'm showing with the black arrowhead, as well as a non-glycosylated species, which I'm showing with the white arrowhead. And the fusion is as well. But interestingly, the extracellular domain mutants, as has previously been noted, are strictly non-glycosylated. And when we look at the distributions of these receptors, they're actually quite different. So the fusion, unlike the wild-type receptor, associates with the cell surface extensively. So it's trapped in a cortical compartment, whereas this EIDS has also been previously noted, is trapped inside the cell. And interestingly, in these cells, only the fusion activates phospho-FRS2 and phospho-SHIP2. Next we put these different mutations into my 3D assay, and I thought the results were quite surprising. So the fusion has a very dramatic phenotype where the lumens are basically completely lost, and the junctions are very perturbed with very punctate E-cadherin. And the fusion has the extracellular domain mutant has a very different phenotype that's more modest, I would say. Where there are some lumens present and the junctions appear more normal, and this is quantified on the bottom left. And when we treat these fusion-expressing cells with an FGFR inhibitor, we can see a nice rescue of lumen formation. And this isn't the case with either an ERB inhibitor or a MEK inhibitor. So I think this is a an example of a proof of concept of how we can use a system to model sensitivities of these different mutations to targeted therapies. So I think, to conclude, I think this was sort of surprising to me that different activating mutations in FGFR have very distinct impacts on lumens, and this, I think, suggests that like receptors that might be expected to function similarly might not when you have a highly sensitive assay like this. And this might kind of stratify patients on those kinds of distinctions. So our working model is that in the control situation, you have very tight epithelial organization, tight FGFR signaling that's locally restricted, and this helps to enforce epithelial architecture. With an extracellular domain mutant, because it's trapped within the cell, it's signaling from an internal compartment, and this doesn't disturb epithelial architecture to the same degree. But with the fusion, because it's on the outside of the cell, this can interfere with activities at the cell cortex, and this could lead to a disruption in epithelial architecture. In some important ongoing directions that I have, first of all, we want to test various mechanistic hypotheses for why these different mutant FGFRs first localize differently within the cell and why they have such disparate effects on 3D organization. And one thing that I'm particularly interested in is that we have mounting evidence that different mutations, the context in which these different mutations arise might cause them to exert different effects on the cell. So whether a mutation arises in a cholangiocyte versus maybe more of a bipotential cell or a hepatocyte seems to affect its trafficking and signaling and gene expression. And we're in collaboration with UN, we're going through massive RNA-seq profiling of these different cell lines to identify potential downstream targetable alterations. One major thing that I'm doing right now is to use a 3D system to phenotypically stratify additional mutations in FGFR. There are a whole host of them, either alone or in combination with co-occurring mutations in cholangiocarcinoma. And I'm also using the system to model the sensitivity of these different mutations to a variety of drugs using in 3D. And with that, I'd like to thank Andy, all the members of the McClatchy Lab, as well as our collaborators, my various committees, and funding, and particularly the foundation for so generously supporting me in this research. Thank you so much.